Hi there, this is Biology Review, and today I'm going to take you through some of the things that you need to review and some tactics to review for your first test. Now, some of you have already taken the tests. Before every test, I'll be giving out these kinds of review videos. In the future, they'll be a little bit earlier in the week in the modules, so that you can review them a little bit more before you actually take the test. So. I'm going to go through a list of topics I think you should pay attention to, but first of all, I want you just to kind of like have a strategy to take this test. So the first thing I would do if I were you is I would go through your notes. So first go through your notes. And when you go through the notes, look at your method in particular, your method/rule, because that tells you how to think about these topics. Also take a look at the summaries. That gets you refreshed on the topics. And if you need to, then look at the connections to your life. That helps you remember this information. So your notes are the first stop. Okay? Then I would go through your workbook. These problems are key problems that might show up again. In fact, sometimes they show up exactly the same on the test. I would also think about what were the problems you had on the quiz, so quiz problems, and I'd like you to try to go back and figure out how to solve them based on your notes and based on your workbook. Now, if you don't understand any of these things, then you've always got the videos to fall back on. So this is like the fallbacks, so some things that you might want to look at. Fallbacks. That should be 2 words. So here are some of those things. You might want to take a look at the videos that I've posted, and you can also take a look at the readings that I've posted. Okay. Now, if you haven't looked at the links that I've sent you, the interactions or the interactives that I've sent you and so forth to other websites that kind of go through the same material, then this is the time to do it, because now you get to review and refresh your ideas of what these topics are. So right now I'd go to the external links, the external sites, the external resources. This is the time to do that if you haven't done it already. So after you've gone through this, go through notes first, then workbook, then think about your quiz problems and how to solve them. Hopefully you've been writing out how to solve them so far. That's the third step. And then go to your external resources. If you get stuck on any of these things, then go back, here are the fallbacks over here, to videos and readings, and these will help you. If you still have difficulty figuring out what you need to do for this test, or these ideas right here, the ideas that are within the notes and within the workbooks and within the external problems and the external resources, then you can always email me. But use me as a kind of like a last resort. Email Mr. Sand. I want to see if you can do this independently. Work for yourself here. You can do this. We got lots of resources here. I know you can do this. Okay, especially for this first test, which should be pretty easy. I'm telling you that right now. I know all kids think, okay, well, Mr. Sand says it's easy. That doesn't mean anything for him because he made it. Well, I will tell you now that so far the average is very, very high. So you can take a little bit of ease when you're going through this material, because it's not going to be so difficult. Okay, so this is how, if I were you, I would go about studying for this final, I'm sorry, not for the final, for this Unit 1 exam. Okay? Those are the steps I take. I go through notes, the workbook problems, quiz problems, especially the ones I had difficulties with, then external resources. And if you get stuck along the way here, then go to the videos, go to the recordings, and then email me if you need to. Okay. So, what are the main ideas, the things I need you to do? This is not going to be a complete review. This is going to be kind of like giving you some ideas to look at in your units, in your topics here. Okay, so the first thing I want to talk about is carbohydrates. So carbohydrates, I'm going to get a different color here. I'm going to look at strawberry. Here we go. Carbs. carbohydrates. Okay, so what do you need to know for carbohydrates? You need to know what they are, so you need to be able to identify them. ID the carbohydrates. So if you look at a carbohydrate, you should be able to say, ah, that is definitely a carbohydrate. What are some things to think about when you're IDing a carbohydrate? Well, one thing is that usually these carbohydrates are in a kind of like a ring structure, meaning that they kind of like loop around on each other. Another thing is that they have lots of carbon, and they've got lots of OH groups, which gives them their name, the carbohydrates. Another thing that would link them together is long chains of these structures, these little ring structures here. So if I give you an example, if I go back through, I look at your, here they are, lipids and carbohydrates. I look at your module here. There's lipids first, and then down below the carbohydrates. Let me make this a little smaller so you can see it. And there we go. Okay, so these little ring structures here with lots of oxygens and hydrogens and carbons, those are carbohydrates. If you link them together, they make these long strings of carbohydrates that we call polysaccharides. Singly, they're just called monosaccharides. They're also known as sugars, and sometimes people just call them carbs. Yeah, so that's the thing you need to know about carbohydrates. Lipids, while we're here, we can talk about lipids. Lipids are these long chain structures with carbons and hydrogens. And at the very end, they have an OH group and an O group like this. They always have this. Lipids are also known as fats. They're also known as fatty acids, and they're also known as triglycerides. I'm just going over an overview here of things you need to know. Yeah, so you need to identify what a lipid is, and possibly what a lipid does. What are some of the functions of lipids? What are some of the functions of lipids? Do we have functions listed here? Some functions? So where? I can't remember if I have something down here. Oh, here we go. They pack together nicely. They release more energy than sugars because they've got more carbons. They also store energy. They can be combined with oxygen and air to create water, so that's why camels have them. They insulate well, like they're good heat insulators. So if you want to survive in the winter, you got to have a nice layer of lipids or fats. And they shock absorb as well. So if you get hit, or animals that have lots of kind of like they need to absorb shock well, they'll have lots of lipids in the areas where they're absorbing that shock. Okay, they act like little springs. Lipids are also involved in the plasma membrane, and we've done a whole bunch of stuff with plasma membranes. You need to know, so we've done carbohydrates already, so let's just go on to lipids. Lipids. So here you need to identify them, and I would also say the functions. So for every one of these macromolecules, you need to identify them, be able to ID them, pick them out in a lineup, so to speak, and tell me about their functions. One of the key functions of a lipid is that it's involved in, here's a lipid, there's another lipid attached to a phosphate group. These little phospholipids are in the plasma membrane, in a cell. Okay? And we drew some cells, or cell membranes, or plasma membranes, or whatever we want to call them. They look kind of like this, where you've got two layers, and these things are moving all over the place. They don't stay still. They're not static. And then you've got maybe like a protein that's in between them that is like a channel protein, and this channel protein can send things through it. Maybe it's gated. Maybe it's involved with active transport. Who knows? I'm just drawing a protein, kind of like an unspecified protein there that lets something through. In this plasma membrane, there is this layer right here that does not like water. In fact, we call it hydrophobic. You need to know what that means, hydrophobic. And over here, we have a hydrophilic layer. Okay? I'm going to go back to this note up here, these steps over here. After you've done the external resources, then you should do what you're doing right now, which is watch the review and anticipate the review. What I mean by anticipate is, when I'm drawing this, you should be able to say, oh, he's going to talk about the hydrophobic layer, and then he's going to talk about the hydrophilic layer. I know what hydrophobic means. Hydrophobic means water-fearing. That means there's no water that's going in it. And hydrophilic layer is where the water interacts, and it's perfectly happy to be around water. Okay, so when you're watching these reviews, kind of anticipate what I'm going to say. Guess what I'm going to say. Pretend like you're the teacher. What would you go on to say next? Okay, so here is a really simplified plasma membrane. There's a protein right there. What else should we talk about? Okay, yeah, this makes up the outer membrane of a cell. So this goes all the way around a cell. Right. For the lipids, the ID and function, also now nucleic acids, of course you need to know the ID and functions for that. So ID and function. Some people had a lot of difficulty with this when they were doing their just their simple workbook. Remember what a nucleic acid is and how that's different from a nucleotide. A nucleotide is a sugar, a phosphate group, and some sort of nitrogenous base, or we just call it a base. It's all about the base. That is one nucleotide. If you string one more nucleotide to that previous nucleotide, then you have a nucleic acid. So nucleic acid is just a string of nucleotides. So if you have two or more nucleotides strung together, that is a nucleic acid. Okay? Of course, the main function of this is related to DNA. What else can we talk about here? So we've got carbohydrates, lipids, and then the final one would be proteins. And we'll put proteins right here. Of course, you know what a protein looks like. A protein is just a string of amino acids. And so look at what an amino acid looks like basically, and know what the functions of proteins are. There's lots of functions for proteins. They do just about everything in the cell. So if ever you have any doubt whether or not, if ever you kind of like are in the position where you're like, okay, so what macromolecule does this certain task? More than often, more than not, it's going to be a protein, because proteins do just about everything. Okay, so proteins are very important, and we're going to go through how proteins are made shortly. You're going to love it. Okay, right. Now, all of these structures here, all these macromolecules, can be put into chains or into bigger molecules with a reaction. So here's a reaction. I'll use green to mean bigger. Clover is what I'm going to use. So, far can we go? All right. So for all of these molecules to make bigger, they go through well, what do you think they go through? They go through a condensation reaction. For all of them. I'll abbreviate reaction RXN, because I'm cool like that. This involves one part of the molecule. I'll just say this is the rest of the molecule over here. One part of the molecule has an H group, and over here it has an OH group. And here's the rest of the molecule. It doesn't matter if this molecule is a carbohydrate, if it's a protein, if it's a lipid, or if it's a nucleic acid, they all go through this same reaction. So this is the rest of the molecule over here. Here's the rest of the molecule over here, but then you have this little H group right here, and you have this little OH group right there. In a condensation reaction, this is what happens. This bonds with that molecule, and this O and two H's leaves. It says ciao, sayonara, hasta la vista. It's an OH group now that floats into the sunset. There it is.